Hi, my name is Vivien Wittstruck, and I'm a junior at Freedom High School in California. This is my presentation for the International Young Researchers Conference regarding gap junctions in regenerative medicine. My goal was to establish experimental approaches in studying cell communication. Cell-to-cell communication is vital for tissue homeostasis, development, and coordinated responses to environmental changes. One key form of communication is through gap junctions, which are specialized intercellular channels that enable the direct exchange of ions, second messengers, and small metabolites between adjacent cells. To establish transfer through gap junctions, a reliable method for introducing membrane and permeant tracer molecules into cells is necessary. The technical challenge lies in delivering such molecules into cells. Conventional delivery methods include microinjection and electroporation. However, microinjection is technically demanding and restricted to a small number of cells. Meanwhile, electroporation is expensive, often results in high levels of cell death, and is primarily limited to non-adherent cells. Some research has been conducted using scrape-loading dye transfer to assess gap-junctional intercellular communication in epithelial cells. However, this approach has not been widely applied to HIPSCs, as these cells are highly sensitive to disruption of their colony, so further development is also needed, with some surfactant cytotoxicity being a major concern. This study seeks to establish methods in studying gap-junctional intercellular communication in human-induced pluripotent stem cells, which are cells that can develop into any type of specialized cell from a skin or blood cell sample, to evaluate how gap junctions contribute to colony integrity and coordinated cell behavior. HIPSCs rely on gap junctions to maintain pluripotency and viability. Many cells, including HIPSCs, survive best within colonies, where close cell-to-cell contact provides essential survival signals. When isolated, HIPSCs are highly sensitive and often undergo apoptosis, making them an ideal model to study how functional gap junctions support colony integrity and prevent cell death. Next is MDA-MB231 cells, a triple-negative breast cancer cell line known for its invasiveness and single-cell growth, to assess how gap junction function may be altered in cancer cells. Metastasis is the leading cause of death in breast cancer and involves single cancer cells detaching from the primary tumor, migrating and colonizing distant tissues. Gap junctions play a role in regulating cell-to-cell communication, migration, and interaction with the surrounding environment, processes that are critical during metastasis. I will now introduce the two approaches used in the study to assess gap-junctional intercellular communication in the HIPSCs and MDA-MB231 cells. Both approaches are cost-effective, not technically demanding, and can be applied to a wide range of cell lines and cell numbers, besides the ones used in this study. For HIPSC colonies, scratch loading was selected because these cells grow in tightly packed colonies. This method mechanically disrupts a small area of the colony, allowing tracer dye to enter cells at the scratch site without the use of chemical membrane disruptors. Because the cells remain in close contact, the dye can then spread to neighboring cells through gap junctions. Scratch loading is cost-effective, technically simple, and particularly suitable for adherent cells organized in colonies. For this experiment, the HIPSCs were provided in a 24-well plate. A negative control consisted of untreated scratch cells. Polar tracer CF-dibiocidin was diluted in HBSS at 1:2, 1:4, 1:6, and 1:8 ratios. Each dilution was applied to separate wells to assess concentration-dependent dye uptake. The control well received no CF-dibiocidin. Since the MDA-MB231 cells are breast cancer cells, which grow as individual dispersed cells rather than in colonies, a surfactant-based loading method was chosen. Since scratch loading relies on disrupting a connected cell layer, it is less effective for isolated cells. Low concentrations of non-ionic surfactants interact with lipid bilayers to temporarily increase membrane permeability, allowing membrane and permeant tracer dyes to enter the cytosol. For this method, live MDA-MB231 cells were seeded in 48-well plates and treated with their respective detergent concentrations. Successful permeabilization was assessed using fluorescently conjugated phalloidin, which can only enter the cell following membrane permeabilization. A negative control consisted of non-permeabilized, untreated cells, both live and fixed. Phalloidin uptake was detected using fluorescent microscopy. Images were taken at ×10 magnification. Figure A shows the negative control. No fluorescence was detected in the untreated control, confirming the absence of autofluorescence. Figures B to E show that CF dye biocidin successfully entered cells along the scratch site and diffused throughout the colony at all tested dilutions, with brighter signals observed at higher concentrations. Signal intensity decreased with increasing dilution, indicating dose-dependent uptake. Fluorescence at the highest concentrations were excessively intense, suggesting that lower concentrations are sufficient for effective labeling. Figure A to B shows negative controls. Figure C is the first positive control in which fit cells exposed to a high concentration of trident X100 showed increased Vivien staining due to enhanced membrane permeability. This was conducted to represent successful permeabilization without compromising cell viability. Figures D to F show cells treated with decreasing trident X100 concentrations. Increased Vivien staining was observed with higher trident X100 concentrations, indicating enhanced membrane permeabilization. However, this was accompanied by corresponding loss of cellular integrity. Figure A shows the negative controls. Figure B to F are cells that were treated with decreasing TREEN20 concentrations. Increased Vivien staining was observed at higher TREEN20 concentrations, indicating enhanced membrane permeabilization. However, this was accompanied by corresponding loss of cellular integrity. Figures A to B show negative controls. Any observed staining resulted from dead cells. Figures C to D depict two tested digitonin concentrations. Minimal variation was observed across concentrations. Notable Vivien staining was not detected in live cells at any concentration, indicating that the membrane was not effectively permeabilized. Vivien staining observed likely corresponds to non-viable dead cells. Little staining was observed at low concentrations of trident X100 and TREEN20. Higher concentrations resulted in dye uptake, indicating membrane permeabilization. However, these treatments caused major cell death. The 0.025% TREEN20 treatment showed the greatest staining with less cell death compared to higher concentrations, but this condition was not sustainable for cells and therefore not viable. Overall, trident X100 and TREEN20 showed similar effects, with TREEN20 resulting in less cell death and greater dye uptake. These trends suggest that while detergent concentrations correlate with permeabilization efficiency, there is a narrow therapeutic window before toxicity becomes limiting. This pattern is somewhat expected given the known membrane disruptive properties of detergents, though the extent of cell death at lower concentration highlights the sensitivity of the cells to membrane intervention. Digitonin treatment resulted in cell death at all tested concentrations and did not permit intracellular dye uptake, despite apparent membrane disruption, suggesting that digitonin is highly toxic to MDA-MB231 cells and is therefore unsuitable for intracellular delivery in this context. This finding was somewhat surprising as digitonin is often used for selective permeabilization in other cell types, highlighting cell type-specific differences in membrane composition and cholesterol sensitivity. Collectively, trident X100, TREEN20, and digitonin are not viable detergents for permeabilizing cell membranes while preserving cell integrity, as concentrations required for effective permeabilization consistently resulted in cell death. On the other hand, scratch loading appears to be a promising method for delivering molecules into HIPSC colonies, as mechanical disruption via scratching facilitates tracer dye entry into cells. Among the tested concentrations, the 1 to 8 dye dilution yielded the most optimal fluorescence intensity, directly supporting the hypothesis that mechanical disruption can enhance intracellular delivery without the need for harsh chemical permeabilization. Lighting may have introduced inconsistent membrane disruption or cellular stress. Additionally, only a limited range of dye dilutions was tested, leaving the optimal concentration for efficient delivery without cytotoxicity undetermined. Future studies should focus on standardizing the scratch loading technique and evaluating lower dye concentrations to improve reproducibility and minimize toxicity. Further investigation is also needed to determine whether scratch loading can be used to deliver functional biomolecules into HIPSC colonies and to examine the roles of gap junctions in cell viability, pluripotency, and differentiation potential. The detergent permeabilization experiments also presented significant limitations. The compounds tested caused substantial cell death at concentrations required for effective dye uptake, limiting the ability to assess intracellular delivery. In addition, only a narrow range of exposure times, compound concentrations, and loading conditions was evaluated, raising the possibility that alternative parameters may achieve better balance between membrane permeability and cell viability. Optimizing these conditions could improve intracellular uptake while reducing cytotoxic effects. Here are my sources. Thank you.